Hi friends, see this I'm going to discuss about the origins of anterior posterior polarity or patterning in Drosophila. See, for your easy way to understand or grasp the genes, I have just simplified it in a form of tabular form. You just have to pay attention to this particular form which I have created. Like maternal effect genes are there. These genes will encode transcriptional and translational regulatory proteins which then differentiates through its syncytial blastoderm. That means maternal effect genes will produce certain regulatory proteins which will either activate or repress the expression of certain zygotic genes. A syncytial blastoderm means nuclei within a common cytoplasm, and the nuclei is not surrounded by cell membranes. That's the meaning of syncytial blastoderm. So here you have to concentrate only on this much stuff, that is the maternal effect genes has capability to produce regulatory proteins which can activate or repress the expression of certain zygotic genes. Now actually there are 4 maternal effect genes, or the proteins which the maternal effect genes produce are of 4 types, and they are named. 2 of these proteins, the 4 proteins are bicoid, hunchback, nanos and caudal. Now I will mention the role of these proteins, what are its functions. 2 of those proteins, that is 2 of the maternal effect proteins, that is bicoid and hunchback, are the one which regulate the production of anterior structures, and the another 2 proteins, nanos and caudal, are the one which enable the production of posterior parts of the embryo. These things you should not get confused. Bicoid and hunchback with anterior structure production, and nanos and caudal with the posterior parts of embryo. That means therefore maternally specified proteins are of 4 types: bicoid, hunchback, nanos and caudal. Understood? These are the things which you have to understand first and foremost. That means for the anterior and posterior patterning of the Drosophila embryo, maternal effect genes will maternal effect genes play a significant role, and these genes will how they will produce regulatory proteins by transcription and translational processes. They will produce regulatory proteins. These proteins can either activate or repress the expression of certain zygotic genes. Now, 4 maternal effect gene proteins they are producing: bicoid, hunchback, nanos and caudal. Out of this, bicoid and hunchback will facilitate the production of anterior structures in the development of those structures. Nanos and caudal will facilitate the production of posterior parts of the embryo. In short, I have given in the last table, that is the maternally specified proteins are 4 in number: bicoid, hunchback, nanos and caudal. Now next, the remaining how these genes get regulated by other genes, I will mention it in the next video. Hope you guys understood this much. Thank you.